it will be fine. But I will just send out extra just so that we catch everyone. Yeah. Well, for the people who are already here, welcome. I'm Tom Nijhoff of Cyto Smarts, and I have the honor today to interview Greg Way about his latest research predicting cell health with image painting, cell painting images. Greg, let's start with you. Who are you?
Thanks, Tom. It's great to be here. Thanks for the invitation. I think this might be the first Cytodata sponsored author Q&A, so it's an honor also to be here representing Cytodata Society. So like Tom mentioned, I'm Greg. I'm currently a postdoc scientist at the Broad Institute of MIT and Harvard. And yeah, I've been working with image data for the past couple of years now. It's a really fun type of data to work with. There's lots of information in images, a lot more than one might think, or maybe exactly as much as one might think if you're used to working with images. But there's still a lot of opportunity to discover new and cool biology with images. So it's an exciting time to be working on these data.
Yeah. Yeah, you already said well, looking at your resume, you have worked with it for quite some time with images, with the cell painting. Are you still excited about the field?
Yes. Yeah, that can be a short answer. Yes.
Great. I think that will make the rest of the talk a lot better. In a second, I will ask you to give a short presentation about your work, and so that all of us are up to speed and to hear more about it, what is written. For the people watching, in the YouTube live chats, you can send messages. I will read them and I will then bother Greg with them so that he will have to answer them live on the air. So yeah, keep them coming. There are no stupid questions, right, Greg?
Yeah, absolutely. Yeah, so, I'd love any question. Bring it on.
It doesn't matter if you want extra clarification about things you think are unclear about the paper or about his talk now. It doesn't matter if you want to go deeper into it, want to look in things for the future. Come with all the questions. Also personal questions?
Sure, yeah.
Well, Greg.
Not sure how interesting that would be, but.
Yeah, Greg, I will now go full screen to you. So the stage is yours.
Okay, awesome. Oh, Tom, you've disabled participant screen sharing.
Oh, details.
Maybe we should have done like a trial run or something first.
Oh yes, only if we had done exactly that yesterday and actually had the same problem, and for some reason. Yeah.
Okay, I see it now. So Tom, I also sent in the chat that link, that URL that you see there. So if you want to post it in the on the YouTube directly in the chat, maybe that would be helpful for people to follow along.
I will.
Okay, so no one can hear Tom. He is talking. He agreed to post the URL that I have here where it shows the slides. So this is a link to all the slides. So I'll just get started. Thanks again. Really excited to be talking about this research. Before I get into it, I think it's worth noting that none of this research would have been possible without my co-authors. In particular, Masha, who's the first author, a co-first author on this work, as well as the senior authors, Anne, Paquita, and Shantanu. So the research was recently published in the Molecular Biology of the Cell, so it's a journal, MBOC. And I have a link here, a direct object identifier or a DOI link here. So if you're following along in the slides and you want a comprehensive survey of what we did in this paper, you can read the paper. It's free access. So the title is Predicting Cell Health Phenotypes Using Image-Based Morphology Profiling. And it's impossible to understand what we did just by reading the title. So I'll kind of break down each of these individual pieces so we can understand exactly what was going on. So the first and most important thing to understand is what Tom was alluding to in the intro, and that's a description of what the cell painting assay actually is. And so the most abstract way that you can think about it is like a color by numbers. So it's essentially like you have a picture of a cell that you take with a microscope. And when you have high enough resolution, magnification in the microscope, you can see individual structures within the cell. So you see the nuclei, you see endoplasmic reticulum, you see mitochondria. And what the cell painting assay does is specifically mark some of these substructures. And so this is what the data actually look like. You're not going in with a crayon and coloring in the individual pieces, but you use a series of unbiased dyes to mark individual substructures within each of the cells. And so what this gives you is an unbiased high dimensional representation of cell state. And it's a really cheap assay. You can quantify millions of cells in experiments for actually pretty cheap, cheaper than one would expect. And so it's a really powerful assay because it gives you this unbiased measurement. But one thing that it does lack is there needs to be some additional information there to help you with interpretation. So you can, depending on how you're extracting and measuring the morphology features of each of these individual compartments, there needs to be some additional layer of information that's there, or it's very helpful to have. And so that's what one of our primary contributions in this paper was the development of this new assay. So this is the assays that we're calling the cell health assay. So it's actually a series of two different panels. In the first panel, we're measuring targeted readouts that are associated with cell cycle. So these are going to be reagents that are going to highlight with different fluorescent dyes, different cells that are in different cell cycle stages. So for example, this one particular dye EDU is going to mark cells that are in S phase. PH3 is going to mark cells that are in mitosis. And then we have another targeted reagent that's going to highlight DNA damage specifically. So this is a pretty unhealthy cell that's undergoing a lot of stress, and there's DNA damage that's just happening. And so this is a direct example, and you can see it's pretty heterogeneous actually, if you look at even a small subsection, a small sliver of the microscope image. And then we also have another panel in which it's actually a separate assay in which we're measuring viability. So this is like whether or not the cell is alive or dead, essentially. And then one specific mechanism by which cells die, which is apoptosis. So in the cell health assays, we are measuring very specific targeted readouts using these series of reagents. We then perform a series of image analysis steps. So that means like actually going from the raw image itself, passing it through a data analysis pipeline in which we're now extracting specific readouts from each of these targeted reagents. And we also do we can do like pretty creative things here. So we can like only select cells that are in S phase or only select cells that are in mitosis, and then measure the DNA damage that's in those specific subsets of cells. And likewise, in the viability panel, we can quantify the intensity of the caspase reagent, which indicates like how much actual, like the trajectory of the apoptotic pathway. Like, is this in late apoptosis or is this in early apoptosis? Is the cell dead already? As well as how much reactive oxygen species is present. So reactive oxygen species are generally considered to be negative indicators of cell health. So if there's high ROS that's present, then it's likely that this cell is undergoing a lot of stress. And so the cell health assays, like I mentioned, are going to provide these targeted readouts from microscope images. And so to take the next step, so what we did was we have the same exact perturbation. So we have a plate of cells, and then we treat them with various compounds, or we actually, in this case, it's CRISPR reagents, but that doesn't actually really matter. In this case, we treat them with specific reagents and then see what happens and measure various things. So we measure cell painting, so an unbiased set of dyes, as well as the cell health assay, which is a targeted set of reagents. And so, like I mentioned, we can then actually capture these very specific phenotypes, like the number of DNA damage or gamma H2AX spots that are in the G1 cells specifically. And so what we then do is train a computer or so-called machine learning algorithm to predict the actual targeted readout from the set of unbiased reagents. And this is particularly powerful because the set of reagents that are targeted are more expensive and typically require a lot more hands-on time to process. Whereas cell painting, we have a lot of legacy data, and it's a lot easier to, I guess, process it. So what we see here is this one particular phenotype that we end up extracting. Like I mentioned, the number of DNA damage in G1 cells specifically, and we train a model to predict exactly what that readout is from the unbiased set of dyes. So this is what I'm showing in this plot here, where the predicted values is on the y-axis, and then the ground truth, which is what we're measuring in the cell health assays for this particular reagent, particular phenotype, shows somewhat moderate predictions. Like it's a pretty good model that you're able to predict the targeted readout. And we know that it's pretty good because of our machine learning framework, which I won't get into in too much detail, but just to note that we have this setup in which we have training data. We we essentially train a computer to predict the actual readouts and then evaluate it on a held out test set. So these data the model's never seen before, and it's able to predict to a reasonable degree. And we're even more confident in this because we introduced this permutation-based approach also, in which we randomly shuffle the features and then make predictions, train the model again. So what we see is a reasonable performance in the train set, but then that performance is completely removed in the test set. So this indicates to us that that 0.51 readout is picking up on some amount of signal. And so again, to remind everyone, we're using cell painting data as the training set, and we're predicting the cell health readouts specifically. And there is one step that I also won't get into, in which we extract morphology features using software called Cell Profiler. And so one of the other particularly exciting aspects of this project is we have so much other cell painting data. So if we have these models, we can train them and show that they perform to some reasonable degree. We can then apply them to a set of new data in which we don't actually have those targeted reagents. So I know, Tom, that you talked about in one of the first cytodata tutorial series that we're starting, you talked about transfer learning. And so this particular point here is particularly relevant. And you can watch Tom's previous video to learn more about transfer learning. But in this case, so we have the cell painting readouts from all of the drug repurposing hub compounds. So these are all compounds that are particularly promising to either are being used in the clinic or in some form of clinical trials. So all of these compounds could be given to patients for the patient benefit. And so that we can then go back to these cell painting profiles from the drug repurposing hub compounds and then try to dissect some of the more fine-grained mechanisms using these models. And so this same model that I showed in the last slide, the number of DNA damage in G1 cells, we can then apply these models to all of the compounds in the drug repurposing hub. And we see that indeed, these compounds, these tubulin inhibitors and aurora kinase inhibitors, end up having really high predictions for this particular phenotype. And we know that this is, we know a priori that the tubulin inhibitors and aurora kinase inhibitors actually do induce this specific phenotype. And if we look at a dose response curve for one particular aurora kinase inhibitor, baricitinib, we see a pretty strong response in accordance with the predicted output. So as you increase the dose of this particular compound, you get higher predictions for this one specific phenotype. And so this is just one readout that we're getting from the cell health model, the predictions of the number of DNA damage spots, which I show here in the test set for the real data, the machine learning model performance in the test set. This 0.51 measurement is indicated by this one particular, the width of this bar here. And so the strength of the cell health assay is that we have many more phenotypes than just the number of DNA damage in G1 cells. So we have other measurements like the live cell width to length ratio, the number of live cells, the percentage of dead cells, the amount of intensity that's in the EDU channel in the nuclei. So these are cells that are in the S phase of the cell cycle. We have a whole bunch of other types of phenotypes that are potentially more interesting, like the number of reactive oxygen species, the number of cells in mitosis. And then the x-axis here is the expected performance on a set of data that the model's never seen before. And so we see that there is quite a large gap between performance. So some of the models are able to perform really well, but then other models, we don't really have a lot of signal that's in them. So you have to kind of calibrate your expectations when you're applying these models to new data based on how much we expect them to actually generalize. So why do we like this approach, and why do we think that it's helpful in some ways? So it turns out that in any application, especially in microscopy, labeling cells with some sort of ground truth measurement is really hard. So most of the time it actually requires some sort of manual annotation. So biologists like actually looking at the pictures and saying like that cell is doing this, that cell is doing that. So the cell health assay is more of a quantitative way to kind of try to sidestep this manual annotation problem. And also just in general, cells are a lot more heterogeneous than people tend to appreciate. So even in a monoclonal cell line where you just have like kind of one cell type that's in a dish and you culture it, they're even heterogeneous within that single population. So cells are cycling all the time in different cell cycle phases. And even in certain cancer lines, you can have spontaneous mutations that will then cause like completely different phenotypes and things that you observe under a microscope. So with this approach, we can potentially isolate certain subpopulations that are lighting up in the cell health assays to perform some sort of targeted profiling. So you can imagine like cells that are all in the G1 cell cycle phase actually are impacted by a drug differently than cells that are in the S phase, for instance. And also once we start to scale up the complexities of the types of things that we're looking under a microscope, we can perhaps identify and isolate the disease cells that are contributing to the disease, versus if there's a mixture, the healthier cells that we don't want actually to respond to drug. We want only the disease cells to respond to drug. And also this approach is just generally helpful to diversifying your understanding of mechanism. One really easy example is cell death. So we know from like decades and decades of research that there are many different ways that cells die. And the way that they've been classically defined is just by looking at them under a microscope. So we know that the signal is there. So with microscopy and these types of approaches, we can start to tease apart some of these specific mechanisms in really high throughput and start to annotate mechanism for each of these compounds or other perturbations that we're studying to get like the morphological context, in addition to some of the other things that are more traditionally captured, like gene expression or some other genomic modalities. So I also just want to highlight here that a lot of all of this research that we performed was done entirely in the open from the beginning. So all of our data we released immediately. So the images are currently on IDR, Image Data Resource, which is a really nice repository for making data easily shareable and viewable. Our single cell profiles, which is an intermediate data type, they're all available on Figshare. We include our entire data processing pipeline, as well as the downstream analyses and the actual aggregated profiles themselves. All of that is on GitHub. And then the drug repurposing hub data, which is the data that we applied our models to, and then explored some of these compound predictions, are also available here at this link. Like I said, the software, so all of the data analysis downstream is done in this GitHub repository. And then also we have this web app, which I'll just quickly highlight, and we'll see if this doesn't completely break in live. You know, people generally don't recommend to do these types of tutorials live, so we'll see what happens. But you can go to this link here, and actually this is really helpful for biologists who can then kind of select their favorite compound and explore the types of predictions that we're making. So this is the site here. There's some images that are about to load. So we have 70 different cell health indicators in total. Some have better predictions than others, like I mentioned here. So these are the predictions here. And then these are all of the individual compounds themselves with their annotated predictions. So this on the bottom, for example, this axis here represents the reactive oxygen species in the cell. And this is the number of G1 cells. And you can see here that these cells that I'm highlighting here are mostly going to be these proteasome inhibitors that are known to have high amounts of ROS and relatively low amounts of cells that are in the G1 cell cycle. And you can toggle this here, so you can look at drugs or you can look at CRISPR perturbations. There's a lot fewer CRISPR perturbations, but we have the drugs here. You can look at the cell health data, or you can look at a reduced representation set. So this is UMAP space. You can toggle the exact phenotype that you want to profile. So this is the percentage of cells that are in apoptosis. And I'll go back to looking at the cell health here. So it looks like the percentage of cells in apoptosis are strongly correlated to the number of reactive oxygen species. And then there's a couple of other like individual plots here where we're looking at dose, if there's any particular dose response for a particular compound, which then I can look at here. And then it shows this dose response curve. And there's a couple of other features on here that I won't get into, but it's there for y'all to explore if you're interested. And yeah, that's what I got. So thanks for your attention and looking forward to if there's any questions.
Yeah, thank you. That's a great presentation, and also indeed thanks to show all the people on there who were helping and participating in this research. I see indeed some compliments also coming in from the audience. Yeah, I'm collecting the questions from both YouTube, LinkedIn, and Twitter together.
Awesome.
Well, but first of all, you already made it quite clear that the open science is quite a central part in it. Did the open science also help you do your research by doing it directly from the beginning instead of releasing everything afterwards?
Yeah, that's a great question. There are definitely different levels of open science. So scientists can choose when to make their stuff open. And I think in general, the field is kind of pushing closer and closer to like immediate openness. And so that's what we chose to do in this project. And it really helped a lot. I think from not only like a career standpoint to like selfishly, I'm able to freely talk about this work, no matter where I am, to no matter who. All of it was open. I didn't need to worry about any sort of like confidentiality or anything. I could just speak freely about it at any point. So that was really fun to get, feedback from lots of different scientists throughout the entire process was awesome. And like, because all of the data is open, we can use that data for more than just one analysis. So we have ongoing work that uses data for various things. So the drug repurposing hub data that links cell painting data in that repository, we're using in like 4 or 5 other projects. The cell health data specifically, we're using in a bunch of different projects, and there's no restrictions on that. So anyone can use it, including us, which is the great part.
Nice. Oh, that sounds great. A question is coming in from Leon. How stable are your results in the cell line holdouts you do? Do you think it would be easily applied to novel cell lines, or that one could allow users to fine-tune their own data?
Yeah, that's a great question, and thanks for the question, Lone. Yeah, a reviewer on the paper actually asked the same thing, and we have an analysis that we do. It's like supplementary figure 13 or something like that, in which we do so the approach that we took was we trained our models using 2 of the 3 cell lines. So that's something I didn't mention. We use 3 cell lines in this data. We use A549, ES2, and HCC44. One's a lung cancer line, one's an ovarian cancer line, and one is a colorectal cancer line. And so it would be super powerful if these models are able to generalize no matter what the cell line context is. So that would be awesome if we saw that. But in our approach, which is what we did to directly answer this question given the data, was we trained our models on 2 cell lines and then predicted on the third. So we're already kind of shooting ourselves in the foot because we have a lower amount of our sample size is lower to make that prediction. So in general, we do see a drop in performance in that approach, no matter which 2 we train on and then predict on the third. But we still do see signal, so better than random, a lot better than random in some cases, for a certain proportion of the phenotypes. And I think these, if I remember correctly, these were the ones that were more general. So this is like, is there G1? Like, is a cell in G1 or is the cell in S phase? So this is mostly to do with the amount of DNA content that we observe in the nucleus. Whereas like some of the other more shape-specific models, like the live width to length ratio, those are harder to predict, just because the exact cell shape is going to be more intrinsic to the cell line itself than it is going to be a generalizable signal. But there's still lots more to do, including collect more data. So the first thing that we need to do is just increase the amount of samples that we're capturing. And then we can actually train even more complicated models in this approach that could more directly account for some of these cell line specific heterogeneity aspects that we think are present.
Okay, thanks. I hope that answers also your question. And I see you also said a second question, so why not? You can retrieve data from existing cell lines, but do you think it is a better estimator of the overall cell health than what you could get from more unbiased morphology profiles? Sorry, I think that's also closely related to the previous question.
No, I think it's slightly different, and is really getting at like the core of what we did in this experiment. So if I understand correctly, the question is like, can you extract these specific targeted phenotypes, whether they're related to cell health or some other aspects, like some other specific targeted biological phenomenon that's happening in a cell? Can you capture that with an unbiased stain? And so we're seeing in this small subset, this small context of this cell health related phenotypes that, yeah, we can use the unbiased signals in kind of like some similar thing to like unsupervised learning based approach to annotate the phenotypes. And so you can extend this research in another direction in which you're not capturing just the cell health specific phenotypes, but other phenotypes as well. But that would in some ways require additional assay design to capture these targeted reagents. And what's nice is that we have legacy cell painting data that we're collecting all the time. And once we have these models, then we can apply them to all these other legacy data sets to continue to extract value from them.
Yeah, no, that is great indeed. If data can be reused and repurposed, that would save a lot of money and research hours. There was something that I noticed about your paper while reading it. We saw the cell profiler from Broad Institute with the cell painting from Broad Institute on the drug repurposing hub from Broad Institute. And of course, you also reference your old papers from Broad Institute. How wide is the technologies and the standardization you're trying to find adopted outside Broad Institutes?
Yeah, I love that question. I think it's a really important one. So I guess it boils down to like, how widely can this approach, how feasible is this outside of an institute that has tons and tons of resources and lots of brilliant scientists. It turns out that there's brilliant scientists elsewhere too. So there's lots of people doing cool things around the world. And there's even people with the capabilities in various labs too. So we know that cell painting is being used around the world. There's an initiative called the Jump Cell Painting Consortium, in which I think it's like 13 pharmaceutical partners are all building up their own cell painting assays in-house, their cell painting programs, and this high throughput, high dimensional microscopy programs. There's individual labs that are doing cell painting assays pretty regularly, and Cell Profiler is being used everywhere, like in labs that are analyzing images. So in small batch experiments as well as in high throughput. Yeah, but that is always a concern. And so we hope that by opening the data up and by opening the code up, that more people can have access to the data and do their own research with it. We're not putting restrictions on what research people perform, what questions they ask with the data that's open. But it is definitely a big consideration for us, that anything that is done needs to be reproducible, no matter what context.
Oh, great. It is, also indeed good to hear that although everything originated from the same points, it really spread out to other places, and that you're not caught, well, too much in your own biases. Let's ask a bit of a general question, but what is the future direction this work will lead to? Oh, for the people watching this, we are still taking questions. All the questions that are sent, I am seeing them live. The difference is you see us talk about 2-3 seconds later, but keep them coming. So to repeat it, so what will be the future steps? What would be the next things that will be needed in order to bring this research to its full potential?
Yeah, there's so many different research avenues that we can pursue next. And a lot of people have been thinking about these types of research questions for a while, and some of these thoughts have been percolating around a bunch of our groups. So it's not just my ideas that I'm about to say, but kind of where the field seems to be headed. I think one exciting research direction is to make these predictions to see if the signal exists in the absence of cell painting reagents. So there's a bunch of different ways to capture cells under a microscope. One of them is cell painting. So cell painting is this set of unbiased reagents that's going to target these various substructures in the cell. But there are other ways that are less traumatic to the cell. So cell painting requires that you fix the cells, and that's, you basically only have one opportunity, one snapshot in time to take these pictures. But you can imagine if these signal exist in some of these less traumatic ways of taking pictures with a microscope, like bright field imaging, for instance, then you can extend this to multiple dimensions. So you can capture live cell imaging over time to see how these particular phenotypes are kind of evolving in a time course. This is also super amenable to more like patient-derived lines or like actual cells that are taken from the patient. So these are all cancer cell lines. And so one really exciting thing is this idea of rapid ex vivo testing of patient-derived lines. So you take a tumor, you grow out the cells, and in bright field imaging, you can have these predictions of these cell health, toxicity measurements, and you can treat these patient lines with a whole bunch of different drugs at different concentrations, and maybe even mix in some healthy cells to make sure whatever you're treating doesn't actually harm the healthy cells that are in this patient. And then basically characterize the impact of the cell as it's going through over time. so then perhaps you could select a specific treatment for one of these patients, given its profile of how it's responding to the drug in real time.
Yeah, that sounds really great. The personalized medicine, you already touched upon it in your paper as well. That's a field, well, I think a lot of us will be excited about. Um, we also have a question from Lassie. I don't know if you by any chance know him. He asked, do you have plans on training general deep learning models for predicting cell health on cell painting images?
Yeah, thanks, Lassie, for that question. I thought for a second there you were going to ask me if I had any plans for this weekend or something, but but yeah, this yeah, that's a great point. One of the major limitations of this project, which I didn't describe in detail in the presentation, but it was a pretty small set of perturbations. So we had only 119 CRISPR perturbations in this, so 357 individual profiles when you account for the 3 different cell lines, so 3 times 119. And there's no way currently to map between the 2 assays at a cell-to-cell basis. So you have only the bulk readouts from the cell painting assay and the bulk readouts from the cell health assay, because there's not a one-to-one correspondence. But it would be super cool, like if we increase the size of the data, and then kind of like you mentioned, kind of overlay in the individual cell painting image, kind of like these individual per cell predictions for these individual models. I'm not really too sure how we would set that kind of system up, but if you have any ideas, I think it would be great to collaborate on. It would be super cool and super powerful at a single cell level, like you mentioned, like in the actual cell painting image, to have this overlay of descriptors plus their expected performance.
To go a bit further into it, you're talking about per cell, but when we're talking about personalized medicine, we are normally talking about per cell sample. Well, by taking a cell sample instead of trying to go for the individual cells, you can take quite some complexity out of the problem, like what recurrent pharmaceuticals did and showed during the last cytodata symposium. Would an approach like that also work in here?
Yeah, definitely. And so we always, at least at the start, like to take the complexity, kind of tone it down a bit, just to see if we are able to detect signal. And that's what we showed in here. So we applied basically like the simplest machine learning model that you can think of, just logistic regression, just to see if there's signal there. And then because we saw that there was signal, then we can start to kind of unravel that complexity a little bit. And so one thing that kind of restricting the complexity and looking at it from a simple problem, one thing that does in this particular context in regards to personalized medicine is that we know that cancer as a direct domain is very complicated. So there actually are these smaller sets of individual cells within these subpopulations that resist treatment. So we know that these cell populations exist. So if you treat a patient with a particular drug, it's likely that it doesn't, like when the drug fails, it doesn't kill all of the cells that are in that tumor, and there's this small subpopulation that survives. And so if we can use a single cell like approach that Lassie had suggested, then maybe we can start to even identify those single cells that might persist after treatment. And that will give us like some idea of maybe we need to use some combination therapy to treat this patient. Or we need to kind of toggle the dose a bit, or maybe we can calibrate the expected percentage, the probability that this particular patient, that this treatment would help this patient for a particular percentage of the cells that we're able to subsample in the dish.
Thank you. I hope that will answer also Lassie's questions in more detail. Yeah, well, to go completely into own bias, There is quite a lot of cell painting data already out there, with and without labels. Could that be used in order to train a generalized model that then by transfer learning could be indeed outperform more of the current scenarios?
Yeah. Yeah, I think that's a pretty similar question to some of the others that we've touched on. I'm not too sure exactly what we would how we would be able to do that at the single cell level, given the data that we have now. But we kind of purposely set the bar really low with this paper. We tried a handful of different models and saw that they kind of all performed similarly. But with even a simple model, we're able to predict decently well. And so now the next step is to take some of this publicly available data, maybe even scale up, collect more cell health images, more cell health assays, and make it open as well. And try now to improve performance, one that to a level in which completely outshines this fairly low baseline that we've set. So I shouldn't completely discount the performance. Like, some of the models actually were very good, but then you could see in that one of the figures that I showed, kind of there's a long tail of models that have only moderate performance and even sometimes poor performance. So if we can increase data and increase our modeling capabilities, then we're almost guaranteed to see an increase in performance for these particular readouts. And also in close collaboration with other, with these folks that we worked with before and other microscopy scientists who might be interested in other targeted reagents, we can even extend the panels basically in the cell health assay and make it more of like a cell state assay. So we could target like very specific features in these cells, and then kind of see if these unbiased stains actually contain more information or contain that information that we're actually looking to target, which would be cool.
Yeah, thanks. I don't see any more questions coming in at the moment. So I think I want to wrap it up over here, or is there a shout out you wanted to do quickly at the end?
I didn't prepare for this one. I'd like to shout out all of my collaborators on this project, the funding sources, which are listed in the paper, the CITO Data Society for giving me this opportunity to speak, the Broad Institute for hosting me during my postdoc, the Carpenter Lab for just being awesome. Yeah, everyone that's in my life. I feel like I'm like giving an Oscar speech or something like that. Like I've
also possible, but a bit shorter is appreciated.
Yeah, Tom, you also shorter. Yeah, Tom, you promised that we would talk about my cats.
Oh yes, absolutely. Before I forget, how many cats do I need in order to replace your work in such a way that your wife would not notice?
Yeah, so far the cats haven't been really good at household chores. Like they're not really good at doing dishes, at least not yet. We'll see, but I don't really have too much hope. In terms of my like computational work, there's that one thing where there's monkeys and typewriters and being able to replicate Shakespeare. So far, we don't know how many cats it would take to replicate the cell health project, but it's good that all of the data is publicly available. So if they have the opportunity, maybe they can. So it should be fairly easy. I have two cats, Mishkin and Fezziwig, yeah, so they're not computational biologists, but maybe someday.
Oh, we'll see. Let's see. They would not even be the first cats to co-sign a scientific paper. You should look it up. It is
yeah, I have to.
A theoretical physicist who did not want to be the only author on his paper, so he made his own cat the co-author. So yeah, we'll see. I will end the live stream here. I will stay in the Zoom call for a second longer. For the people watching, thank you. Um, Thank you for the questions and, of course, for your attention. This talk will be online afterwards, so you can watch it back. You can play Greg on twice the speed if you want to, or half the speed if you think he is talking too fast. But yeah, no, thank you very much. And also let us know if you have other authors with interesting papers that you think should deserve more attention. Well, goodbye.
Bye.